Welcome back to Day 0 to Breakthrough. It's day 107 and I'm joining Diego and Connor to pan for something more valuable than gold: antibodies. The team are on the hunt for antibodies that can bind to virus. I asked Diego to explain why this is an important part of the process, especially in light of the disappointing pseudovirus assay results.
So the best result would be to prove that the mice get protection against the viral challenge. The second best result is to have antibodies that can help us further down the process.
Antibodies are produced by immune cells called B cells, which are found in the spleen and lymph nodes. The B cells the team have extracted from their preclinical studies contain a mixture of naive, inactivated B cells, and hopefully, some B cells that bind to the virus viral fusion protein. Over the past few days, the team have been sorting their B cells out into single cells and extracting the mRNA that codes for the antibodies. Today, I'm watching as Diego sets up a reaction to convert the mRNA into DNA, which is a lot more stable. Diego explains what he's going to use it for.
In a nutshell, we will just take the information that encodes for the antibodies that are specific for our antigen and make it like isolated from the big pool of information contained in a cell. So we will isolate and purify DNA that is specific for that antibody. Then we will transfect it into our cells, produce a lot of the antibody, and test if these antibodies bind to our antigen and even if they are neutralizing.
It's all well and good to show that our vaccine antigen produces an immune response in mice, but what about in humans? Connor, who we met on day 71, has developed a technique to experimentally produce human antibodies before clinical trials begin, using a method called phage display. I caught up with him to have a chat about what this entails. Phages, or more professionally, bacteriophages, are a kind of virus that only infect bacterial cells. To do this, they have their own version of a viral fusion protein. What Connor can do is use some clever genetic manipulation to replace this viral fusion protein with various naive human antibodies.
And we can create a library of these bacteriophage with millions and millions of different combinations of genes to sort of recapitulate an entire human's immune response. And so then we can use that platform to then basically screen for any antibody combinations that are going to bind to our antigen.
This is helpful for the pressure test and future pandemic responses, as this technique can show the team that human antibodies are capable of producing an immune response to the target virus, in this case, virus. These antibodies will also have some important uses further down the vaccine development pipeline. I asked Connor if he learned anything important about the phage display process through the pressure test.
Probably get started earlier with the phage display, I reckon. So have these antibodies even faster and have them as soon as the antigen is ready to go into mice. We can start also then doing this process, which can take like 3 to 4 weeks to get from beginning to having an antibody, which sounds like a long time, but is actually very, very fast.
If you were observant, you may have noticed that the 100 days goal has passed and we don't have a completed vaccine. While disappointing, this is not a failure. The 100 days goal was incredibly ambitious and has never been achieved before. What the team are learning through this pressure test is immeasurably valuable and will help them come closer to achieving the goal of 100 days next time. For now, the team are committed to continuing moving forward and coming out of this pressure test with a completed vaccine. Before we wrap up today's episode, I wanted to end on a more hopeful note and give you a glimpse into the joy that comes with doing science. Here's one last thought from Connor on his experience during the phage display process.
There's science, I have this term, which is not my term, but I describe it as a specific science joy when an assay or something works, and when 3 or 4 weeks of work that you've done culminates in one ELISA plate, for example, and it lights up, and it's just the best feeling. And you're oh, that's why I do this job. Yeah.
Tune in next time as I join the National Biologics Facility once again to see how their large-scale production of our vaccine candidate's going. See you next time on our journey from Day 0 to Breakthrough.